Okay, we want to look now at vegetative propagation, which is asexual reproduction. Asexual reproduction means there's only one parent plant. It doesn't involve gametes or fertilization. So asexual reproduction is reproduction with only one parent. You're required to know an example of plant asexual reproduction from a stem, root, leaf, and bud. Okay, so learn one example from each. Okay, so these stems are modified. They have a special purpose, and their purpose in this case is for reproduction. So modified stems, we take strawberry as an example. Okay, strawberry plants are capable of producing these horizontal stems called runners or stolons, horizontal stems that run across the surface of the soil. Uh, if they touch the soil at a node is a point where leaves are growing from the stem. If they touch the soil at a node, then we'll have new roots forming like this. These new roots will become established, and eventually the link between the new plant that forms over here and the parent plant can be broken. So this stolon can be cut by the gardener, or it will wither away over the winter months, and you'll have a new plant that has formed very close to the parent plant. This is called a runner. Okay. In modified roots, some roots are capable of swelling with food. Photosynthesis from the leaves sends food down to the roots. The roots swell as the food is stored. So in this case, we have the roots of a dahlia plant. And all the little roots, it began as a fibrous, shallow root system. All the little roots have swollen with stored starch. Okay, these allows the plant to survive over the winter. Now, of course, each one of these roots is capable of developing into a separate plant. So we could have a new shoot developing uh, from each one of these swollen roots. So we get the following year, we get a cluster of new plants right beside each other. Okay, so these are swollen roots in dahlia, and these are known as root tubers. In modified leaves, some leaves, when they fall onto soil, are capable of sprouting new plants. So in this case, these are begonia leaves, okay, and they've fallen onto the soil, or a gardener has placed them onto soil, and you can see little new leaves are beginning to grow. New plants, rather, are beginning to grow from the edges of the leaf. Okay, so these will all develop into new plants. Okay, so it's a modified leaf. Begonia is the example. And modified buds. A bud is an area of new growth. Okay, if we look at this example, onion bulbs. So this is an onion. So it has a flat stem and a bud, and the bud is surrounded by layers and layers of leaves. These leaves are swollen and they store food. Okay, but if you examine the little flat stem at the base, sometimes we have two or more buds can develop. So this means that this is planted, then two separate onion plants will grow from this. So there's the main bud and a smaller bud from the side. Okay, so these are the leaves that store food, and the outside of the onion will have a dry leaf, a scale leaf for protection. So what you need to know, examples, just to be able to name an example: strawberry runner, a dahlia root, a begonia leaf, and an onion bulb has a modified um, bud. So more than likely you could be asked give two examples of vegetative propagation in plants. Okay. Now, which is the best method, reproduction by seed or reproduction by vegetative propagation, to be able to make a comparison? So in a comparison, we're going to look at advantages and disadvantages of both. So laying your work out like this is a good way of presenting an answer in the exam. So if you look at reproduction by seed, what are the advantages to plants using seeds? The main advantage is it produces more variation. So pictures of the different variations within a single species of flower. Good example of all the differences, all the variations you can get. Some of these flower colors could be more adapted for attracting particular insects than others, for example. So it produces more variation. This leads to evolution and adaptation to new habitats. Why do we get variation in reproduction by seed? Because it involves sexual reproduction, and sexual reproduction involves gametes that are formed through meiosis. So sexual reproduction and meiosis both lead to variations. It allows dispersal because part of the reproduction by seed processes that seeds are going to be dispersed. So that means it can move to new locations and get away from the parent plant. The disadvantages, it's slow. It takes almost a whole season. It's a complex life cycle that has a number of steps, from pollination to fertilization to dormancy to seed dispersal. And it often depends on external factors such as the wind or insects for pollination. So they can be listed under disadvantages. Now to look at vegetative propagation. The advantages, it's only needs one parent compared to the complex two parents required over here. It's fast. It produces clones or identical new plants very quickly. And it keeps the good quality of parents. So if the parent had a good colored petal or large fruit, then all of the daughter plants will have the same good qualities because they're clones, they're genetically identical to the parent. The disadvantages, okay, because they're clones, there's no variation, so there's limited opportunity for evolution or adaptation to new environments. No external factors required. I suppose that should be an advantage. That should be up there. No external factors required should be up there. New plants often overcrowd parent plants due to a lack of dispersal. So we do get crowding in reproduction by vegetative propagation. Here's the example of overcrowded daffodils. It's great that they all have the same color. They're all brightly colored, which is an advantage, but they're overcrowded. So that's going to affect the growth of all of them because they're crowded for space. So that's a disadvantage. Okay, and finally, we need to look at 4 methods that gardeners would use to artificially propagate plants. Okay, rather than letting nature propagate or reproduce the plants. Artificial propagation is a reliable process that produces faster new plants, keeping the qualities of the parent plant. So it's a reliable process. You can be guaranteed result. It's fast. Okay, you get new plants very quickly. And you keep the qualities of the parent plant. So if you had a rose that was a nice bright red color, then you would take cuttings so that all the next generation of plants are exactly the same. They're clones. Okay, so these are 4 examples, and you're required to know 4 examples of artificial propagation. So first of all, we look at tissue culture. So this is quite a modern method. Hasn't been around that long. So plant tissue culture is used to grow plants from cells under sterile conditions on a nutrient culture medium. So if we're starting off with individual cells of plants, so you can get many, many thousands of new plants from one individual parent plant. It produces clones of a plant in a method known as micropropagation. So it involves an AR, it involves plant growth regulators, starting off with a single cell. The correct regulators are applied so that it develops into a new plant. Okay, so this can be mass production of many plants from one parent plant. This might be used to propagate plants that are difficult to grow, plants that have a very long life cycle. So it could be used to propagate rare plants. In cuttings, it's quite simple. There's a plant, you cut a piece of the stem from the plant. If you cut at a node, this is an area where leaves come from the stem. Nodes are areas of the stem where there's a lot of meristematic activity. There's a lot of cells that are dividing. So you tend to have new growth possible when you cut at a node. Dip the cutting in rooting powder. Okay, that's going to contain a growth regulator that stimulates root growth. And planting in compost. The new plant, the cutting will grow into a new plant. Okay, so you can take many cuttings from one parent plant, and all of the new plants you get are clones. They're genetically identical, the same features. In grafting, the stem of the plant that you want to grow is spliced or placed onto a root of a different species of plant that's already established. So you might, for example, have an eating apple tree, and you can cut stems from the eating apple tree, which is very slow growing, and attach them to the root of perhaps a wild apple tree, which grows very quickly. So you end up with a combination plant, a fast growing root from one species, and the stem would have a slow growing stem from another species. The advantage there is that you can get fruiting apples, or an apple tree that produces fruits very, very quickly, rather than waiting 20 years, which would be normal for an individual apple tree to grow. So you can end up with fruits being produced much sooner. Okay, so grafting is splicing or placing the stem of one plant onto the root of another plant. Okay, the stem is called the skin and the root is called the stalk. And finally, look at layering. Okay, when stems that are attached to the parent plant bend down, so the gardener can peg them down like this. And if this is a node is an area again where a leaf would have developed. You'd have a lot of meristematic cells there that are capable of dividing. So in this case, the plant will produce roots in this area that can become established, and a new plant can be staked up and prepared here. So this method is called layering. Okay, so you have to be able to name and maybe write a simple sentence about each of these: tissue culture, cutting, grafting, layering. And the likelihood is you'll be asked to name 2 methods of artificial propagation in plants. Okay, so if you were to go back on this little section, you could be asked to describe 2 natural methods of vegetative propagation. Okay, so sorry, to back a slide, if you could be asked to pick 2 methods of natural propagation and then describe 2 methods of artificial propagation. So that would be 4 things to learn there. Okay.